Good afternoon, everyone. I think I can actually sit down in 5 minutes because I was planning to introduce us, and I think Tracy actually did an amazing job. So she gave my talk for me.
She made us sound really awesome.
I know. The reality is that I didn't find a job, that's why I decided to not. But yes, I've been taught to not to talk too fast and not talk too slow. So I'm just going to go. Melvin, as Tracy said, Malvindar and I studied together. Actually, we did our PhDs under Sarah Richard Hall, and we did it in Huntington's disease. And that's why we're very, very close with Huntington's disease is very, very close to our hearts. And it doesn't matter what we do, we always come back to Huntington's disease. And that's why I actually didn't want to only study medicine. I wanted to do something more. And that's why I'm still researching about Huntington's. So I'm going to talk, I think a lot of what I'm going to talk about is going to be familiar to those people who've been here last year, but it's always good to start from the beginning of the story, I guess. I think I did something funny. Oh, okay. Sorry. So I'm just going to tell you guys, again, you've seen this before, those of you who've been here last year, but this is where we work, across the road from Auckland Hospital. It's very pretty outside, and I think they're just trying to make it look a bit less daunting with lights and stuff like that. So it's pretty nice looking. And then you've got this beautiful lab. Everything's white, it's quite open, so different researchers, they work together, we actually share our stuff together, we share ideas, and that's the whole idea of open plan. So compared to probably what you used to see 20 years ago, which was little rooms where scientists used to sit and look down the microscope, I think nowadays in world-class laboratories you see that it's open planned, and people share ideas as well as equipment, so that's us. So I'm going to talk about the main goals of the Center for Brain Research, also known as CBR. Unlock the secrets of the brain, and by that I mean we look at different diseases, Huntington's, Parkinson's, Alzheimer's, and what we want to understand is how these diseases actually work and what happens in the brain of these patients. Which then, so this is what someone like Malvindar and I do, and then we introduce or we basically feed whatever we find to other researchers who come up with report pharmacologists. So they actually come up with new therapies. So they look at what we find, and then they say, okay, now I'm going to come up with these drugs for you guys. So we've got a lot of these people also in our center. We also have doctors across the road at Auckland Hospital, neurologists, neurosurgeons. They actually work with us. So whatever we find, we talk to them and we said, these are the things that we found, and then they are going to improve their clinical care according to what we find and what we tell them. And of course, we train scientists and clinicians. So for example, Malvindar has a few students that have come to the center, and they train under us. And clinicians, clinicians like doctors who want to actually understand a bit more about research, they come and they actually spend maybe 3 months to a year with us to get trained in terms of being scientists. So they want to understand how research works. And I think it's very, very important because a lot of the times scientists get very, very detailed, and sometimes clinicians actually don't understand how science works. So it's important to have that connection between clinicians and scientists, and that's how we can come up with drugs that work, and that's how we can actually understand what the patients want. Engage with our communities and educate and inform, and that's exactly what we're doing today. And that is a very, very important role that we play, and we are very privileged actually to do that. I'm pretty sure I talked about the Neurological Foundation of New Zealand Human Brain Bank and how important it is in our research and how proud of it we are and how privileged we are to work at a centre that has a human brain bank. As I said before, I think again last year, we are one of the very few countries to have a human brain bank. I think, if I'm not wrong, 5 or 6 or max 10 different countries that have it, and New Zealand is one of them. And that's why we are actually targeted by a lot of, not targeted, but a lot of researchers around the world want to work with us because they don't have this resource. So we are extremely proud of it and extremely privileged to work with human brain. So, since last year, not much has changed, but again, it's at the centre we actually have Huntington's disease cases, Parkinson's disease cases, Alzheimer's, motor neuron disease, and also very, very rare diseases such as Pick's disease, frontal temporal dementia. We have researchers who work with stroke. So we have different cases with different diseases. And we have different researchers that work with these. One thing that's very, very important is that we also need normal brains. So these are the brains that actually don't have any neurological diseases. And the reason for that is because for us to understand what's going on in a diseased brain, we need to compare it in a normal brain. So that's why even though 227 normal brains might sound like a big number, it actually is not, because for every single study that we do, we have to go back to a control case, again, a normal brain. So not only we need disease brains for our research, we need normal brains. What makes our research special? We're not talking ourselves up. It's real. What we think makes our research special is the Neurological Foundation of New Zealand Human Brain Bank. It's the fact that we work with human brains. It's very, very different when you look at a rat brain or a mouse brain, a monkey brain even, to a human brain. So we are looking at the disease and at real time, and that's very important. So this centre, or brain bank, has actually provided us with the opportunity to start to study the brain in the human brain, to study the disease in the human brain, compared to a lot of cases where you study the disease in animals or you study the disease in dishes, which we will talk about later. But here, you're actually studying the disease as it is going on. So you're basically watching a movie. The ultimate gift. So I cannot emphasize on this more. The most invaluable gift to science, basically, is the brain. However, we are not very selfish. We do actually share our resources with other people. So we do have researchers around the world who approach us, and we do collaborate with them. So a lot of the times, if, for example, a researcher in the States say that they found this drug and they really want to work with the human brain, we do part of their studies for them, and we collaborate for them with them in order to make a bigger picture of their studies, using the resources that they probably don't have, but we do. Simplifying what it actually is, but one of the things that we do is we look at normal and sick brains, as I said before, and what we do is we color them with different stains, and then we look at their structure. Sick brains or sick cells compared to normals. How do they look? How the branching looks. Do they look smaller? Do they look bigger? Are they there? So look at cell loss. Look at the structure. Look at changes in color. Sometimes, if, for example, they're lighter than normal cells, it means that they're losing some specific proteins. So these are the type of studies that we do. We also do grow new brain cells. So I don't know if we talked about it last year or not, but every single brain that we receive, provided that we don't receive it too late, we actually, what we can do, we can grab a piece of that tissue, so we can grab a piece of that brain, put it on a dish, and we can feed it. So we feed it with different type of sugar, for example, and we put it in this incubator that has the perfect temperature. And we make sure that the pH is okay. So we are basically babying that piece of tissue. And it has these cells that even though the donor has died, that piece of tissue still has cells living in it. So by feeding those cells, we are growing the cells. So they keep, if you want to say, they keep multiplying, or new cells are being born. So you start with 1 or 2 cells, for example, and then new cells are being born because the mother cells are being fed, and then at the end you end up with 1,000 cells. Why is it important? Because when you discover a new drug and you want to test it on something that's very close to human but not new human, a lot of people go into animal studies and they try it on animals, but a lot of these cells are not going to be the same in animals and human, so it's important to put it in these cells. So for example, a new therapy is developed, and what they do, they give that drug to us. We will put it on these cells and see how they react. Do they shrink? Do they get smaller? Do they get bigger? Do they survive better? Do they die? So these are the different things that we actually look at when we are. So these are very, very important when it comes to therapies. And I guess we are one of the very few people that do this. I actually don't know of any other brain bank around the world that can grow human cells from tissue. In case you need to know a bit more about how to donate brain, you need to talk about with Marika Asach, who is our Human Brain Bank Manager, and I'm sure we can leave her phone number and contact details with Tracy. Yeah, so Tracy has them. However, again, it's a very, what we do is, what she does is a very, very case-to-case basically basis. So she makes sure that everything is fine in terms of are there other neurological diseases that might, so there are different donation criteria that she will talk about when she's approached. Alright, so now into some exciting research. So I think what I'll talk about is some of our research that Malvindar and I have done, and then I think Malvindar will take over and she'll talk about some of the new therapies that are, I think, very, very exciting therapies that are being now tested around the world, 3 or 4 of them. 4 of them. So I think I'll spend a bit less time on this, and then Malvindar will pick the therapies. But I think it's very important to start with this because what we do is we are trying to understand how and where brain cells are lost. Now, we're not talking about brain cells. I might sometimes refer to them as neurons. So these are the main cells of your brain. So these are the cells that basically let you talk and let you walk. So these are called neurons. And both Malvindar and I work with Alzheimer's disease and also Huntington's disease. One thing when you're looking at these, it's basically both of these are human brain and they're cut coronally, which means this way. So if you cut the brain this way, this is what you're looking at. In Alzheimer's disease, you've got you've got healthy brain over here. Sorry? So it's not showing up. Okay. So you've got healthy brain over here, and you've got a Alzheimer's disease brain over here. Again, they've been cut this way. It's very, very easy to see how Alzheimer's disease brain basically has been affected everywhere. When it comes to Huntington's normal, so this is normal and this is Huntington's, okay? And again, this is a very, very severe case of Huntington's disease where you can see that the whole of the brain is basically affected. But as I will talk about a bit later, I think in the next slide, in our lab, when you're talking about Huntington's, you can see that for a long, long time, people actually thought Huntington's is actually a single region disease, which means that it's only that area of the brain is affected. I'm going to refer to that area of the brain as the striatum. So it's that little area of the brain in the middle of your brain. And you can't really blame them, because if you look at, again, I feel like I keep using that, but if you look at your normal, you can see that that area is very, very big and juicy and nice. When you go to Huntington's, you can see that it's shrunken. So it's very, very obvious. Whereas when you look at the rest of the brain, it's not as obvious as something like Alzheimer's. So for decades, people actually referred to Huntington's disease as a striatal disease. So that means that only the striatum is affected. However, for I think it's been 10 or 12 years that our lab has decided to look at other areas of the brain to come up with new therapies, because what we saw in Huntington's disease actually did not match up. That area of the brain alone cannot be responsible for all the different symptoms that you see in Huntington's disease. And that's when a lot of researchers that were specifically under Sir Richard Ford, decided to look at different areas of the brain and come up with basically a better understanding of what's going on in the brain. And still today, we have not mapped out the whole brain. So still today, we don't know exactly what's going on in different parts of the brain in Huntington's. So, in Huntington's disease, you've got a dramatic loss of tissue in the striatum, again, that little area of the brain. However, as I said before, you cannot explain the variable symptoms that you see in Huntington's disease, because different parts of the brain do different things. And that's why, again, we decided to go to different parts. And that was one of the things that I did during my PhD, looking at the cortex. Now, what I mean by the cortex is that big area of the brain, so it's the outer area of your brain. That's called the cortex. And different parts of the cortex is going to do different things for you. Okay, so there's one part of the cortex that is going to help you to move. There is one part of the cortex which is the frontal cortex. That's very important for your behavior. So if you see someone's behavior is changing, and a lot of the times you hear people with neurological diseases that they say, for example, my grandma's not the same person, it's because that area of the brain is affected. Okay, so that's what we're trying to do, building up the pieces of the puzzle to understand the HD circuit. So we challenged the idea that Huntington's disease is a single region disease. We went around the brain, we looked at different parts of the brain, and every single year, I guess, I can't say every single day because our research takes ages, so every single year we come up with new understandings. Okay, so we are part of a team spearheaded by Sir Richard Ford, and we've basically uncovered different patterns. So one of the main things, I guess, that our group has come up with is that if you look at the brains of different Huntington's disease patients, you understand that they're actually losing cells, or different areas of the brain are losing cells differently. So you see some patients losing cells in that area of the brain that's important for behavior. You compare it with another patient, and those patients are not. They're actually losing cells in that area that's important for movement. So that's basically what we're doing, and that's why we put that puzzle in there. We're trying to put things together and see why is it that different people lose cells in different parts of the brain. But what we found out is that it has a direct link with their symptoms. So you might have two siblings that come with the same gene, but one of them is having behavioral problems, and the other one's having motor problems. Why? We don't actually know. Why is it important to know this? It's important to understand that Huntington's is a whole brain disease, because when it comes to therapies, targeting a single area is not going to help. You actually need something that can target the whole brain. You need something you can deliver to the whole brain. Because I think before, a lot of people treated Huntington's so that they can, if they've got a drug, they will go to that area, the little area of the brain that I talked about, the striatum, they will put it there, and they were hoping for a miracle. But now we know that that therapy wouldn't work because you're just targeting a small area. What I'm going to talk about is, next is this area of your brain called the cerebellum. And I'm going to talk about this, I think, because both Malvindar and I are very, very excited about these results. This is one of our final studies that is, I think, we haven't published it yet, we've just sent it for publication. But this area of your brain, which is right at the back of your brain, is important for movement, coordination, balance, and posture. So I'm standing here, I can hold my posture because my cerebellum is working. And that area of the brain is actually that first area. If you drink too much, that's the first area that gets hit. And that's why you see drunk people can't actually hold their balance. A lot of people ignore that area of the brain. The reason they ignore it is because it's very, very complicated. It's as small as it is right there, but it actually has 85% of your cells. So most of your neurons is compacted in that little area of the brain. That kind of shows how important it is. But I think it's very hard for us to understand it, and that's why we usually ignore it and put it to the side. So for, I think till now, people thought that, or they wanted to think that it actually didn't change in Huntington's disease. They ignored it in Huntington's disease, despite the fact that we do see a lot of problems with balance and posture in Huntington's disease patients, but nobody actually decided to study it. Too hard to study. But what we decided to do, we decided to map it out. So I'm going to talk about, before I get into too much detail, I'm just going to tell you this is the control. So this, okay, sorry. This is your control. Okay, so both the top and the bottom. So by control, I mean those brains that do not have any neurological diseases. Then you have HD mood. So by HD mood, I mean those patients who donated their brains and they basically suffered predominantly from behavioral changes and cognitive. By cognitive, I mean they had learning difficulties. What else? When you talk about cognition, learning and memory. When you're talking about motor, so these are the two motor. So these are motor. So this is the two panels are from a motor case. So this is the patient. These are the patients who suffered from motor. So anything to do with movement, chorea or rigidity. So these are the things that we classify as motor. Okay, now if you look at these big cells, these are the main neurons of your cerebellum. So they're quite massive because they've got a lot to do. They talk to each other. As you can see, they've got these spaghetti kind of branches that are going up. They talk to each other. They talk to the rest of the body. They talk to the rest of the brain. So they've got a lot to do. They're called Purkinje cells. It's not very important, but that's just their name. What you can actually see is if you compare your control with your mood cases, you don't see a lot of Purkinje cell loss. They look very, very, very similar. So those patients who suffer mainly from mood symptoms, they don't lose cells in the back of their brain, cerebellum. If you compare, on the other hand, your motor cases, so this one, so these are the patients who suffer from movement disorders, with your normal, you can see how drastic the cell loss is. So this is telling us that those patients who suffer from motor disorders, they're actually, in Huntington's disease, they're actually losing cells in the back of their brain, or the cerebellum. Now, why is it important to know this? Again, it's very, very important when it comes to therapies. You can't ignore different parts of the brain. You need to come up with something that can target everything. So again, this is hot off the press, and we are publishing it now, so if you talk about it with anyone, we have to come and grab you and basically send you to jail. I was in jury duty, I wasn't going to say. This week I spent a lot of time in jury duty, so I wasn't, no, not jail, no. Just give you a little talk. All right. Where is that research going now? So that was basically an anatomy, what we call anatomy, which is understanding the structure of the brain. That's anatomists. That's what anatomists do. So they sit there and look at different parts of the brain. And I think what we are doing now, we're trying to combine our understanding of the structure with pharmacology that I talked about. So drug development. That's what we're trying to do now. However, before I think that's drug development as well. Mel is going to take over. But before we talk about that, I just want to say that we know the pattern of made brain cells, neuronal cells. Now we've got a very good understanding of it and how it's linked with different symptoms. But what we need to understand again is that brain is not that simple. You don't have just neurons sitting there. You've got those big juicy neurons, but they need to be protected. They need nursing. So that's what every other cell in the brain does. So you've got these little cells that are going to help these big cells survive. They're going to protect them. It's kind of like the rest of the body. You've got your immune cells that protects you from a virus. So it's the same thing in the brain. Your brain has its own immune cells. So we are going to talk about now these cells, and the reason we're going to talk about these cells is that these cells are now being targeted for therapies. So a lot of people have identified that, okay, your neurons are dying, but it's not because they're not getting nutrition, it's probably because these small cells are not doing their job. So the nursing cells are not being good nurses. The immune cells are not mopping up rubbish in the brain. So we're going to talk about these. Bear with me, it might be a little bit complicated, but we might try to make it as easy as possible. So you've got the human brain. Oh, I'm not going to use this, sorry. So you've got the human brain, and the human brain is being, it has a lot of blood vessels. So it's getting a lot of nutrition because of the blood vessels. Now, every blood vessel has those little cells that we talked about that are very, very important when it comes to making sure that rubbish or toxins don't get to your brain. Okay, so we're going to very briefly talk about those cells. So you've got these yellow cells over here. They're called endothelial cells. They make up the tube of your blood vessel. So their role is basically sitting right next to each other to make up the tube. And then you've got these two different types of cells. They're called pericytes and smooth muscle cells. What they do is that they make this cocoon around your endothelial cells. So they're basically sitting there, hugging your endothelial cells, And the reason that they do that, they're basically trying to make sure that toxin does not cross your cells, your endothelial cell or your blood vessels to get into your brains. Then you have your astrocytes, so these guys over here, and also your microglia, so the green one over there. What do? If there is anything that is like a toxin or it's rubbish and it gets to your brain from your blood vessel, these guys are like vacuums, so they pick it up. Okay, if it's too hard, just thinking about it this way. So they together, I forgot to say that, together they actually make this barrier called the blood-brain barrier. So all these different cells together make the blood-brain barrier. And if you look over here, you've got your neuron that is sitting here, sending its processes down to your blood vessel and basically getting oxygen and sugar. So that's what they eat every day, oxygen and sugar. The rest of this, so the blood-brain barrier, is going to make sure that neurons don't eat anything else but those. So if there's rubbish in the blood, your blood-brain barrier makes sure neurons don't get it. That is if your blood-brain barrier is intact. Think about it this way. So you've got Mexico here. Mexico is your brain. Then you've got the States on the other side. Okay, so the States is the rest of your body. Then you've got this wall. This wall is made out of bricks. Every single brick is one of those cells that we talked about. So those cells, and this guy, Trump, is our toxin. Okay? So what we are trying to do is trying to protect our brain, Mexico, from the rest of our body, which has Trump. Okay, now, if that didn't get, so we've got the brain, the pink, and then your brain does not need whatever your body is getting. So your body might, for example, you take paracetamol, your brain doesn't need paracetamol, your body does. So the blood-brain barrier makes sure that paracetamol does not get to your brain, for example. I'm sure everyone knows about paracetamol. Alright. Okay, I think I'll just leave it there and Malvindar will take over.
So Nasim just summarized like 25 years of research. Like seriously, in those slides, 25 years of research. And that bit on the blood-brain barrier is now the hottest area in research at the moment, not only for Huntington's, but for Parkinson's, for Alzheimer's, for stroke, for almost any neurological disease. Why? Because you can tell it's a barrier. There's something wrong with this barrier in these neurological disorders because our neurons are not being protected. So all those cells that Nasim talked about, they're currently the focus of almost like every research group in the world at the moment because we don't know what they do. Yet, the single neuron, which is that main building block of our brain, has been studied, and excuse my pun, but it's been studied to death. And that's partially because they've been dying. We know they've been dying. But what we now need to do is protect them. And so this is what Nasim essentially got to. So you study the brain and the pattern of neuronal loss for, Nasim said about 15 years or so. That was up with Sir Richard Fall. And now we're trying to move to these other cells because we know at the end of the day, you can't protect something that's dead. Right? You need to stop it from dying. So this is why we're now turning to the blood-brain barrier to protect our neurons from dying, essentially. So some of the ways in which we're studying the blood-brain barrier in Huntington's disease involves, so I've got my model here demonstrating this machine. And this machine here is called a tissue microarray. So you know how we've spoken about the brain bank, very precious resource. We want to maximize the tissue availability. And this machine here is a way for us to take 2mm pieces of tissue, align them up in a gorgeous array, and test for different proteins that are thought to be affected in the blood-brain barrier. So for example, we find a protein that's been thought to be a potential drug target in an animal model of Huntington's disease, which is what the majority of the states looks at. Trump is over there, they're studying animal models. And so these guys are, they've found this protein. If you generate a drug to this protein in this animal model of Huntington's disease, the animal model is cured. And we're like, okay, BS, right? So what we do is we find that protein using these tissue microarrays and see what relevance, first of all, they have to the blood-brain barrier, because that's what everybody is saying is affected in these animal models, and also whether they could be a potential drug target. So one of the facilities that Nasim and I are in charge of now is the tissue microarray screening facility, and this is one of the ways we've been studying the blood-brain barrier in Huntington's. I'm actually going to present some of that data to you today. This data is unpublished, okay? So it means that we pretty much are putting our lives on the line in front of you guys and sharing stuff that the rest of the world doesn't really know. So very quickly, what is a tissue microarray? So a tissue microarray is essentially a flat piece of wax that contains little tiny pieces of human brain tissue that we have used a cylinder to extract and align up in a gorgeous array. Why is that really cool? Because it means that the brain doesn't get wasted. It means that the rest of that piece of tissue that we've taken a little biopsy from is spared and can be used for 10 million years from now. Might I add that there's not a single brain in our brain bank that we've actually depleted. So it means that as we continue to collate brains, we're able to compare data that may have been extracted from a brain from 3 days ago with brains from 30, 40 years ago. This is the power of this kind of technique. And so this is an example of a Huntington's tissue microarray that Nasim constructed, yes. And the blue are control brains, so they're all normal brains. And the red are Huntington's brains. So what you can pretty much do is you can make an array like this last about 100 experiments. So we can study 100 different proteins of interest in the blood-brain barrier at the same time. And in this case, because we have 28 controls and 28 Huntington's brains, we basically have a screen of nearly 60 brains that we can look at the same time. It's pretty cool. So some of the exciting data that we've got so far, might I add, this is not published, it's not even in the media, there's no world-famous publication yet, so you're getting this hot off the press, okay? What we've found, and let me just, Nasim did a great job of explaining the panels. This is a snapshot of a control brain, and this is a snapshot of a Huntington's brain that we've been looking at under the microscope. And this is the data we've collected from one of those tissue microarrays and looked at about 60 of these, okay? So these are just the snapshots. Remember those supporting cells that help take care of the tube, the blood vessel? We found that there's a certain protein in these cells that goes up in the Huntington's brain. Drug target number 1, okay? What we've also found is that this is a close-up of a brain cell, which is, remember how Nasim was talking about the immune cells of the brain? This is a close-up of a normal one, okay? This is a close-up of the Huntington's one. Can you see the difference? Now, I don't know whether the branches of that cell are broken, or whether the branches of that cell have gone in, right, have gone into the cell, and it's formed some sort of structure which is immobilizing it to fight battles. Okay? So this is a very exciting thing to look at. Might I add, we discovered this using 2 millimeters of human tissue. Okay? So that is ridiculous. Now, all of us are going to be looking at drugs to target these cells. Okay? These immune cells called microglia. The other thing we noticed is, you know the cells that make up the tube? The yellow ones that Nasim was talking about? What we found is that there are more of these tubes forming. So in Huntington's, there's a bit of a debate in the literature about whether the blood vessels, so the blood-brain barrier essentially, is breaking or intact or changing. Okay, so changing could be a good thing, changing could be a bad thing. What we can say so far is that it's definitely changing. What we can infer is that the blood-brain barrier is trying to make up for something. So more of these spaghetti-like structures are forming in the Huntington's brain. Now, is that to deliver more oxygen and nutrients to the neurons that are vulnerable? Right? We don't know. But what we do know is that it's now a drug target. Okay? Because it's something that we can change in the disease. So what we've essentially presented to you, right, is that this is just a real quick snapshot, but basically it's saying is that the blood-brain barrier is changing in Huntington's disease. We now have 3 potential drug targets. We're going to pursue these and look at these in different models. But we also have, from an anatomist point of view, 20 million other questions, and some of them involve, okay, so we know that these cells are affected to differential degrees, but you know that bad protein, that Huntington protein that accumulates in neurons and causes them to die? Do these cells have Huntington protein in them? That is our next question. And fitting into the story that we've told with the neurons, that we know that the pattern of neuronal loss links with the type of symptoms in Huntington's, is the pattern of these guys changing also linked with the symptoms? Are we essentially now explaining why some people get some symptoms over others using these cells as well? So linking it in with what happens with neurons. And also, is this happening throughout the brain or only certain parts of the brain? This is something we need to know, because when it comes to developing drugs, right, we need to know where to give those drugs. So knowing where these changes happen now is really critical. We only looked at one region of the brain for this work. Okay, so now it's like opening up 20 million cans of worms, which is great. Now, Nasim already touched on it, but one program that we're now starting next year, and I'm really fortunate to actually be funded to do this work, we're going to be testing we're going to be studying the cells that we've been growing from post-mortem Huntington brains now, because we've now generated enough samples to be able to do it. So remember how Nasim said that you can take a piece of tissue that's essentially dead, has a few living cells in it, and cause those cells to keep growing? We now have enough to be able to compare the Huntington's brain cells that are living in a dish with normal ones. So our drug screening program is going to start next year. And I'm really fortunate to be able to actually carry this forward for the next couple of years. This is super duper exciting because a lot of people, the way they study the impact of drugs in a dish, right, is they use cancer cells that grow really fast and multiply really fast to test the impact of a drug. What we have here are Huntington cells with the genetic mutation that have come from a brain that had Huntington's that we can now study later on with different drugs. So this is going to be the work that hopefully by next year, so when we come back again, we'll be able to present preliminary data on this work. And to give you an example of how interesting the Huntington cells are that we can grow, these, I don't know if this might be a bit difficult to see, but do you see the splodges all on this panel? Do you notice how the splodges are just kind of chilling out, not doing much? Okay, so those are just an example of some Huntington cells. But what happens is when we expose them to a toxin called ethacrynic acid in this case, do you see those black dots? Okay, the tiny black dots, they form in these cells. So what we know is that these cells behave differently in response to different toxins. So what we can then do is, okay, can we get rid of those black dots, right, by putting something else on there? So that's going to be the next step in this line of research. And another way we're studying the blood-brain barrier in Huntington's disease is using a machine called an RNA scope. So we all know Huntington's is a genetic disorder. One of the difficulties with studying gene-related changes in a disease is that in order to extract the DNA, to study the genes, you have to destroy the tissue. You have to mush it up to release the DNA from it. we now have this really cool new technique called an RNA scope, which enables you to study genes in tissue when it's intact. So you can look at individual genetic changes in cell types while they're in tissue so that you know whether X cell has a different genetic makeup to Y cell. Whereas before, you didn't know which cell the genes came from, the genetic changes. So we're now going to be using this cool technique to study Huntington's disease. And this is really, really important because when I talk about the drugs that are coming out, and so you're going to hear about all the different drugs that I think are important for you to know about, a lot of drugs are taking, they're taking advantage of the intermediate phase between when the gene creates that bad protein in Huntington's disease. The intermediate phase is called the messenger phase. Okay, so you have the protein, you have the gene, the intermediate is here, it's called the messenger, and this technique enables you to study the messenger in different brain cells. And that's super duper cool because at the moment everyone's developing their therapies based on only the genetics in the blood. Okay? But we need to know more about the genetic changes in the brain. Because what we do know is that there are subtle genetic changes that are different in your blood, to your brain, to your skeletal muscle, okay? But we don't know the magnitude of those differences. So that's what this technique is going to be doing as well. So that's some of the exciting stuff that we're doing at the moment, and that's in progress. Now what I'm going to talk about is stuff that I don't do. But what I've done is I've read about all the massive trials, and I've essentially given my opinion on them. What are the ones to really pay attention to? And I haven't done this alone. I've consulted a lot of people. But what are the ones to pay attention to? And what are the ones that are still in progress that you should just keep an eye out? Okay? So the first one I'm going to talk about is the drug trial that's currently being powered by Roche and its little baby company, Genentech. This drug is the Huntington lowering drug called RG6042. Okay? Currently, this drug is in what we call a phase 3 study. Now, what is a phase 3 study? Basically, a phase 3 study is to test whether a drug is safe and effective in Huntington's disease. And so, this drug was formerly known as Ionis HTTRX. So when you're doing your research, RG6042 and Ionis HTTRX are the same things, okay? So when you're doing your Googling and you're reading about these drugs, they're not different, they're in fact the same thing. Just that one company took over from a little company's work. So the clinical trial is going to involve monthly visits to the clinic over 25 months, and the drug is going to be delivered via a lumbar puncture, so a spinal tap, into the lower spine. And there will be a few other assessments done every time you go and visit for your shot. So Genentech, or Roche, they've spent a lot of time over the past couple of months identifying the sites around the world where this phase 3 trial is going to be taking place. The first official announcements of where these sites are going to be in the world is going to be either at the end of this year or very early next year. Some sites already know that they're candidates, other sites don't. Nothing's online at the moment at this stage. I know as of October 2018 there was nothing there, and as far as I can see there's nothing there online yet. But rumour has it that hopefully Australia and New Zealand will be included in this phase 3 study. And at the moment, from what I understand, there's quite a few talks around the ethical approval of these trials and all that sort of stuff, but we just have to wait. Because at the end of the day, there's no point doing a drug trial unless all necessary rules are in place. So this, at the moment, is the most exciting news to date for Huntington's families. And as a scientist, I'm not even saying that to bring hope or bring despair or whatever you want to hear from me. This is the biggest thing since the discovery of the gene in '93. And the reason why is because it's a targeted therapy. And I'm going to now explain why. So why is this drug the coolest thing since 1993? So, who knows how this, does anyone know how this drug actually works? Okay, would you like to know? Okay, cool. So this drug is known as an antisense oligonucleotide, or an ASO, okay? So in the research community over the past 5 years, it's been like ASOs versus CRISPR technology. These have been the two arms of trying to how to treat Huntington's disease, ASO versus CRISPR. And so what the hell is an ASO? Basically what it is the best way to describe it, I think, is to understand how the gene creates the bad protein and where this drug works to stop the creation of the protein. So it's a tiny piece of DNA. Okay, that's what an antisense oligonucleotide is. And it can actually freely enter the cells, and you can program it to do whatever you want it to do. So in this case, the ASO has been designed to destroy a piece of the messenger that creates the bad protein in Huntington's. So the way to explain that is, that's our DNA. Everybody knows the DNA looks like a double helix.
Yeah.
From the DNA, right, we get different proteins being made that help our body do things, okay? Helps our body create pigment for our eyes, helps our body break down the meat that you had for dinner, or whatever you ate, right? There's different proteins. In this case, what I'm talking about is this strand of DNA that helped create Huntington protein. All of us have Huntington protein, okay? We need Huntington protein to live. We need Huntington protein to multiply our cells in our body. We need Huntington protein for our metabolism. We actually need Huntington protein for survival, which is why we couldn't cure Huntington's disease by getting rid of the protein completely. Because what happens when you do that? You die, okay? This is what has baffled scientists since '93. Yay, we discovered the gene, but oh shit, you need it to live. So what they did was they knocked out the gene in animals. The animals didn't live beyond 3 days. So then scientists were like, shit, okay, what are we going to do? We have to turn down the production of this protein. We have to kind of like use not the mute button on the remote. We need to lower the volume on the remote so that you get just enough to live, but not enough to get Huntington's. That's essentially what scientists have been focusing on. This therapy is a way to turn down the production of that protein. And the way it does it is this antisense oligonucleotide, which is this piece of DNA, binds and destroys a piece of the messenger, right, that creates that protein. The caveat to this drug is that it doesn't know the difference between mutant Huntington protein and normal Huntington protein. So then all the scientists were like, oh, damn it. Right, so if we turn down Huntington too much, right, what's going to happen to people? So then what they did was they tested the drug on everything. Cells in a dish, rodents, larger animals than rodents. So then they went to primates. And what they could see is that this antisense oligonucleotide doesn't work at 100%. It works at about 70 or so %. It means it turns down the volume, right, of both the normal and the bad Huntington protein, but it doesn't completely turn either of them off. What that means is that even some of the bad Huntington protein will still be made, but hopefully not enough to kill neurons, right? So that's what we're trying to do. That's what this therapy is trying to do. And these people have been working on this therapy since the early 2000s. As a scientist, I've been hearing about this therapy in conferences every single year, and I've been like, yeah, right, okay, so what? It worked in a rat. Yeah? And then I'm like, yeah, right, okay, now it's working in monkeys. Hang on a minute. And then I'm like, wow, okay, this is something that's actually looking quite promising. And then everyone was like, oh, this could actually make it into a human. So, what they did was the scientists that were working on this antisense oligonucleotide partnered up with a company called HDYONZ. And what they decided to do is in 2013, they created this big deal about the fact that finally we had a drug that was going into a clinical trial. And in 2015, they started the trial. And who did they start the trial on? They started the trial on 46 heroes. We call them heroes now. They started the trial on 46 people in Germany, Canada, and the UK. And the trial began in 2015, and it ended in November 2017. So shortly after that, all of you would have seen in the news in December of 2017, this headline. Phase 1/2A study in patients with Huntington's disease has a dose-dependent reduction of mutant Huntington protein observed. Did you guys see that? Many of you would have seen that and been like, what the hell? Like, what does that actually mean? What that pretty much means is that, remember how the drug's delivered via lumbar puncture? When you give somebody a lumbar puncture, you can also take a little bit of their fluid in their spine. Mutant Huntington, that bad Huntington protein, is present in the spine too. And what they found was that the more drug you give, the less mutant Huntington protein you see in the spine, in the cerebral spinal fluid. This was a big, big, big deal. And this is why the drug has now been accelerated to a phase 3 clinical trial. This is why they're now testing it on hundreds and hundreds of patients across the world. But this is not the only Huntington lowering drug in town, and this is very promising, because you know what happens when Big Pharma gets involved? A fight happens, right? So this is what's happened. 3 other companies have now developed different types of drugs. One of them's called weve, the other one's PTC Therapeutics, and the other one's Voyager. These are all companies now to look out for, because they've all developed different Huntington lowering programs. But before you get too excited, one thing I want to say is that they're all at different stages and they all have different ways of delivering the drug. This is good, because now we have competition. And this is also even better, because remember, based on Nasim and I's research, patient A with Huntington's is different to patient B. We know this. That means drug A may not work for patient A. It means drug A might work better for patient B. The more competition we have, the more combination therapies we'll also have. If you think about HIV, for example, it's not one drug that treats HIV, it's actually a cocktail. Okay? So this is really super duper exciting. So Wave's approach is basically what they're saying, Weve, is that, so they developed an antisense oligonucleotide, but it also affects the normal Huntington protein. We're going to try and develop something more specific, but our company's going to do it. So that's what Weve has essentially done. So what they've done is they've taken advantage of the fact that some people who have the bad copy of the gene also have other small gene mutations around the Huntington gene. And there's 2 specific mutations, they're called SNPs, single nucleotide polymorphisms. And they've developed 2 more of these antisense oligonucleotides that target the 2 SNPs, the 2 signatures around the mutation. Certain populations of people with Huntington's have these 2 particular mutations. So what they're saying is that, if we create this messenger-killing antisense oligonucleotide that's specific to these 2 SNPs, we're going to get the Huntington's patients that all have that mutation, and therefore they're going to respond to therapy better. Okay? So what they've done is created, I wouldn't say a better version of what the Roche company is doing, but a version that might benefit certain populations better. So this is now getting towards individualized therapy. This is a company to watch out for, Weve. Okay? So I've mentioned at the bottom, in a perfect world, targeting only the mutant Huntington gene would obviously be better. But we're not living in a perfect world. We're trying to figure out what makes the mutant gene different. That's why we're studying the genes in these cells. Okay? Another drug, PTC Therapeutics. This one currently hasn't reached humans. Now, might I mention the Weve drug has reached humans. That's phase 1/2A. So we hope to hear from them in 25 months' time whether that drug has been beneficial to the small sample of patients they've been working with. So that drug's gone into humans as well. This one isn't. This one's currently being tested on rodents, but I'll tell you why it's important. How cool would it be if we didn't have to have a lumbar puncture to treat Huntington's?
Yes, that would be good.
That would be cool, right? How cool would it be, right, to take a pill to treat Huntington's? Now, I'm not talking about take a pill to treat the symptoms of Huntington's. I'm saying take a pill to treat the genetic problem with Huntington's. So this company called PTC are developing things called small molecules. Okay? And these small molecules, what they're claiming is, remember how I said those antisense oligonucleotides are pieces of DNA that get into cells? These small molecules, apparently, are molecules that will get into cells, but they cross the blood-brain barrier, so you take it in a pill, it gets through into the brain, and it attacks the messenger the same way. Now, before, scientists were like, we smell bullshit because this is just science fiction. This is what we've dreamed of. PTC Therapeutics have claimed that they've got this successfully working in rodents. Now we wait and see. That's another one just to watch out for, just to have in your Google tab. And finally, Voyager. Now, Voyager are using a different tactic. Remember how I said at the beginning that there was the team of the antisense oligonucleotide, and then there were the teams using CRISPR. Okay? So now I'm coming towards the gene team, which is more like CRISPR. So Voyager is using gene therapy. Now, this type of therapy is currently being tested on primates. So it's gone to bigger animal models, and when it starts going to primates, you know it's something to look at. This is, okay, when you're searching for drugs for Huntington's, when it says it works in a rat, just go boof, ignore, okay? When you see it working in a sheep, in a pig, or in a primate, then you're like, okay, I'll save this search for later. Okay, so this one, what it does is it uses a virus, okay, a single injection of a virus. And the virus has been deactivated, not to give you the cold, to give you HIV instead of Huntington's. Like, it's a deactivated virus that has no bad properties, and that virus, inside of it, has a little packet of gene therapy. Okay? That packet of gene therapy then gets released into the brain, and what it does is it stuffs up the production of Huntington protein. And the cool thing about this therapy is you only need to have one injection. So far what they've found is by injecting this gene therapy using the virus into primates, I'm sure it's primates, just double check it on the line. Yes, monkeys. Monkeys. So by a single injection into monkeys of this gene therapy in a virus packet, they were able to knock down the production of Huntington protein, which spread quite far in the monkey brain and stayed. Because what this therapy does is it hijacks your own machinery and keeps creating the drug yourself. So this is an example of kind of using your own immune system or using your own genetics to fight the disease within yourself. So now we have not just one therapy, but actually 4, many of them using slightly different mechanisms to treat Huntington's disease, and they're all in different stages. 5 years ago, we had nothing. 3 years ago, we had nothing. Now we have something that's made it to phase 3. And one thing I do know is that the patients, that the 46 people that were using that ASO, right, have not got worse. That's real cool. Because at the end of the day, these drugs, okay, these drugs aren't going to be that beneficial for someone who's got millions of brain cells already dead. Okay? This drug is going to be useful for someone who's at the earlier stages, not necessarily showing symptoms yet, but might be gene positive, right? Who wants to slow down the production of this bad protein and slow down the death of their cells. Which means, hopefully, the idea is that the onset of Huntington's would be very late. That's the whole point of this drug. So the take-home message, if you ignored everything that I talked about for the past...
I have a question about the last drug.
Yes. Yeah.
When do you think that it will be tested on humans?
So based on the history of the first drug that I was talking about,
Yeah.
the data they released on primates came out about 2 years before they started testing it on humans. And the reason why is to get ethical approval from the FDA to be able to put this drug into humans involves a lot of hoop jumping. But when they look at that data from primates, that's something that gets taken real seriously. But they don't just take that at face value. They want to know about primates that have lived for 3 months with the drug, primates that have lived for 6 months with the drug, primates that have lived for a year with the drug. So when I say primate data, that data has been collected over years to get to that point. So normally to get it to straight through to a clinical trial, it's normally a year to 2 years. Hopefully the race will make things easier, because now that kind of paperwork that's been involved with starting a Huntington's trial has happened more than once. So hopefully getting it through those hoops would be easier. So the take-home message about the drugs is that there are currently 2 trials to look out for, not 1, there's 2. The first one is the one by Roche, okay? And the second one is the one by Weve. Currently the one for Roche has passed the test for safety, but now they're testing for effect. And you guys both know, like you guys know and I know, to test for effect, we have to wait. We have to wait for these patients to age, right? So this is essentially what we're waiting for now. The other drug, Weve, is currently being tested for safety. And so that's the 2 different stages. These are the ones you've got to watch out for. But behind that, there's Voyager and PTC. So keep an eye out on these drugs as well. And this is super duper exciting. Last time we didn't present anything on drugs because we had nothing to show you. We actually took the time to read through all of this data and actually give you something that's meaningful, because I don't want to give false hope either, because I'm not out to do that. But what I can say is that these drugs are being tested extremely vigorously. And I've seen the data, and what I do know is that the patients aren't getting worse. Okay? I'm not saying that they're getting better either, but I'm saying not getting worse is a good thing. We have to wait for them to age. And the other thing is that before, scientists were fixated on just trying to treat the symptoms. Now we're trying to treat the cause. So that's what's super exciting about these drugs, is we're going to the genes, we're going to the messenger that creates the protein. 10 years ago this wasn't possible. It wasn't. And as I mentioned earlier, perhaps like HIV and diabetes, these therapies might be the way to go. It might not just be that this ASO works for this person. They might need an ASO mixed with a small molecule, or a small molecule mixed with gene therapy. So this is really cool, because it means that we might get individualized therapy. Because our research suggests that it's not going to be a one-stop shop fix. It could be more than one. So how to stay up to date? Now, rather than waiting 1 year for us to come back and give you an update, one really good source, and there's no, Trump doesn't give any news to these guys, right? No fake news here. One really good source of information is HDBuzz. Who's heard of HDBuzz? Okay, I'm really concerned only 2 people have. 3, and they're all the people that look after you guys. So okay, so HDBuzz, you just put it in Google, and it will give you the latest in the drug trials, and it's written in everyday speak language. And that's really important, because I hate boring science. And if you want to get first dibs on a drug, right, don't cry and moan if you haven't heard about it, if you don't know about Enroll-HD. Enroll-HD gives you first dibs on any information to do with drug trials here in New Zealand. You don't know about Enroll-HD and you've not been in touch with Enroll-HD, do it now. Like now, right now, on your phone. And if you want to talk to people involved with Enroll-HD, there is an Auckland site, but I'm so happy to hear there might be a Waikato site. Tracy, that news has made my day. That's amazing. But until that happens, one person you can contact is Lisa Fraser, and or you can just go to the Enroll-HD website and you'll be directed through to the Auckland-based team. But I'll be so happy if there's one here. How much time do we have?
We've got 5 minutes.
Sweet. Okay. That's great, because all the hard stuff is now over. And so, if you have any questions about the sciencey stuff, please ask us, because we can go over it in more detail. The part I'm going to talk about now is really digestible. It's easy. I think even Nasim over there will get it. Yeah. And so we're going to talk a little bit about HDYONZ, and thanks Tracy for introducing us as members and founders of HDYONZ. Who are we? Well, we are a group of people, there's a few of us, that are dedicated to supporting, educating and advocating for young people impacted by Huntington's here in New Zealand. We report to the big sister international organisation, HDYO International, but we think that it's really important for Kiwis impacted by HD to get Kiwi support as well. And so I'm really fortunate that there's this brave woman who's way more intelligent than I am, called Leanne Knox. And I understand some members of her family are here in the audience today, and so they're representing her. Leanne and I, thank God we get on really well, we co-chair this organisation, and actually Nasim, Leanne, myself, and these people here, apart from the boy at the bottom, he's our newest co-op, we founded the organisation as well. And the lovely lady at the top of the slide, Dr. Samantha Murray, she's our Christchurch representative, so she helps us liaise with the families in Christchurch. Nasim gives us our clinical and scientific advice, and she's hopefully be our future doctor on board. And Leanne has a huge task of representing HD families, and that's like a super duper big deal. Like it's really hard to do that. So she keeps me in check, tells me what to do, and we together have formed this organisation so that we can help other youth out. And so we've partnered up with a lot of different people, but just to really quickly summarise, we're affiliated with the Centre for Brain Research because we're Centre for Brain Research, Brain Research New Zealand, the HDYO International team, but also the Huntington's associations of New Zealand, and that includes MS Waikato. So if you want to see who all the people are that we're in contact with, check out our website, which I will now show, hdo.co.nz. hdo.co.nz. And we tell you about Kiwi research because you can go to the international website to find international research, but we tell you about what's happening in New Zealand, and I just love this tagline. We do a lot of work on the sheep, transgenic Huntington sheep. There's an article posted up there. Have you heard about the latest breakthrough by New Zealand scientists? So we have an article there about the Huntington's transgenic sheep project. We also have a full summary for those who didn't attend the National Huntington's Disease Conference. We have a full summary of what happened at that conference, and we have direct Twitter update links to every single researcher, so you know exactly what they spoke about. Please check out our website. Get lots of useful information from there. What else have we
to that, Malvindar?
Sorry?
Just you saying about getting lots of useful information. A young boy we know did a project for school about Huntington's, and he found that site the most useful site.
Yes.
We referred him to it.
Yes.
He said it was one of the best sites for getting information.
I'm so happy to hear that. And shortly after we created our website, HDYO International updated theirs because they were like, oh, the Kiwis have done it again. I'm so happy to hear that. Thank you so much. But apart from updating our website, which is our way to engage with the youth community online, we've done a few TV stints. So if you haven't seen our pieces on Huntington's in both the Sunday program and Both Worlds, I highly encourage you to watch them. We also do adult meetups. Now I did say last year we were going to make it to Waikato. We haven't, and I'll tell you why, because not many people from Waikato have been in touch with me. To bring me here, you have to get in touch with me. So this is something I'm going to say to Tracy. You've got a handful of young people, we either meet halfway, or we come here, or we bring them up, perhaps for a Centre for Brain Research tour or something like that, whatever you think sees fit. And we want to meet up with the Waikato youth as well, because it's not that far, we have no excuses to get around here. But also, I'm going to talk about the money side of things. We spend a lot of time trying to fundraise, and I'll tell you why. We don't want to compete with the local associations. The local associations rely on funds from lotteries, COGS, and all these things that are, involving grants, and we don't want to compete with them. We don't want to put the same application in with them, because at the end of the day, we're all here for the same purpose. We try and use alternative ways to fundraise for our activities. We also are now we have actually got a really good relationship with Enroll HD and with the genetic counsellors in Auckland, but it would be great to do something like that here. Perhaps we can talk later, Tracy. And who's heard of the Australia-New Zealand HDO camp? Now, many of you might have heard there isn't going to be one next year, but do you know why? That's right. So the reason why we're not having one next year is because HDO said they don't have enough money to fund a camp for next year because they're concentrating all of their resources into the big 2020 HDO International Conference. And so we tried to fundraise a bit of money to have enough resources to be able to do the Australia camp, but at the end of the day we need to focus and concentrate our resources on the conference as well. And so this is just an example of some of the TV stuff we've done. Oh, I just saw you now. Look at you. Look at you. Look at your cute daughter. Oh, celebrities. Oh, Dr. Mehrabi. And so we have a lot of amazing people who participated in TV stints with us. But our biggest missions at the moment are to raise money for a Huntington's youth worker. We've raised about 10K so far, and this is in conjunction with me being a normal scientist trying to do my job in the lab, Nasim being a medical doctor, Leanne holding a full-time job, Sam holding a full-time job. All of us are volunteers. So the money we're trying to fundraise is it's almost like what we do when we should be sleeping, okay? And the other thing we're doing at the moment is we're going to work with HDO International, we have been, to bring as many young people from New Zealand to the conference in 2020 in Scotland. Okay, so that's like our big mission at the moment.
Can you also talk back to how old do you have to be a young person?
Yeah, I've had mixed feelings about this. I think anyone who's not symptomatic. So if you're 35 and under, I think we should still be in touch, because you're still having those same issues as you were if you were 20. You're still, the issues are, should I get tested? The issues are, how do I have kids? The issues are, how do I cope with my family? The issues are exactly the same. And it's the same for a person who's 12. And so I think at the end of the day, we can't put a cutoff on age. I'm happy to talk with anyone who's under 35 and not showing symptoms. And so the last thing I want to say is that we have every form of communication possible, and it's managed by us. Most importantly is our website. Second is Facebook. If you want to see any event that we're doing, check into us via Facebook. And I really just want to make a shout out now to two important people in the audience. Mary Hiron in the back and Peter Knox over there. Because these guys helped us start and help us keep going. These guys are the ones that still share our content, they still promote us out in the community, they're still fundraising for us, and if it wasn't for people like them helping us out, I don't know how we'd be able to do things. So just big ups to both of you, and to all the other families that are actually reading our stuff and sharing the work, because at the end of the day we want to connect with you. And so if we can't connect with you, then why do we exist? Right? So thanks to you guys for keeping us going and keeping us humble and keeping us upbeat. If you want to get in touch with us or get involved with us, there is a form, it's a contact form on the website that will then be sent directly through to us. So please get in touch with us. Thank you all for listening.
So much. Well, there's no doubt about it. It's some really exciting stuff. I can't believe what you presented tonight. We meet just only 12 months ago.
I can't believe that we did it either.
Just the difference.
Nasim was reading some of that content for the first time.
So aren't we very privileged? Okay. We are very privileged to hear some of the stuff that has not even made the headlines or gone into print or anything, so aren't we special?
Don't tell anyone.
Right, thank you so much. That was great. I've got gifts
Oh.
for you.
No, you didn't do that.
So, thank you so much. We know that you guys are really busy. Super. Oh, as I sat down, Peter told me that I missed something. of one of the awards.
Oh no.
Facebook
Dr.
stalker.
Malvindar just got awarded on Sunday for the, you correct me, I wrote it down, I forgot. Kiwi, I wrote it down.
Kiwi Bank Local Hero
Local
of the Year.
Of the year.
Seriously, I just want to say that, like, every time I get an award for something, I feel like such a fraud. Like, and I'll tell you why. If I felt like I had, like, cured something, or, you know, really broke that barrier, then I feel like I'm an actual hero. I think the only thing I'm really doing is just having a louder voice so that everybody can hear what you guys are going through. That's really it.
But you've got to remember, every time you get an award and
It's
they mention
the Huntingtons is out.
it's putting it out.
Yeah, I mean, that's probably the biggest thing.
Yeah.
And I felt a bit bad because, like, I went up and gave my speech because everyone had to give us, I didn't even know. Everyone had to give a speech. And I totally, all I did was talk about the youth organisation. I just plugged for the Huntington's Youth Organisation. I didn't even say what Huntington's was. I didn't have time because they gave us 1 minute. And so I just went blah, 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 Huntington's Disease Youth Organisation of New Zealand. And then I was just, if someone heard me and they visit the website and they get an interest in HD research from that, then it's a win for us. Yeah, so thank you. Peter!
We've put out some food for supper, so help yourselves. Please feel free to mingle and have a chat with these most amazingly passionate researchers that we have. That we're so lucky to have you in our country, New Zealand. So yeah, and also Ron's there to have a chat with, and Liz and Karen and I, if you want to come and chat with anything about any issues, whatever. But thank you anyway. Thank you again.